for it.
Okay, all right. Okay, everybody. Thanks so much for coming. Really appreciate you guys taking the time. Yeah, so like I said, I was just hoping to get a conversation going, just lay out what I got for you guys and make sure that it's okay with everybody, and to see if you guys have any input or suggestions. So so yeah, so I'll just lay it out just in a nutshell, really. So I'm planning on having 4 chapters. Chapter 1, it's I think it's looking in pretty good shape. I addressed most of the comments that that Mark and Will had, and so it's mainly a lit review of all the what's currently known about the small hive beetles. And I met with Ethel the other day, and I added a little bit of I added a little segment on experiences that we've had with the small hive beetle in Hawaii, just working with it locally, just some considerations. And then
mostly like management, practical stuff. Like this is the brown encounter.
Mm-hmm. And then
how many pages did you write for this chapter?
I think it's at 20 pages. Yeah.
And has Ethel read it?
We were working on the latest editions, which is I haven't read his version of the latest editions because that just happened this week.
You have gone through
I have been going through it. I saw the comments that you guys had on it, so thanks. And I think we just basically, especially for the literature review, it becomes kind of an old-fashioned exercise unless you can ground it in something that is useful to Hawaii. So one of the things we wanted was to be able that if somebody references, there's some feedback about what we did from a practical standpoint, because it's talking about control and things like that. So we have chosen to go with a particular method of control. So we were saying, like, from the most basic practical things, like we use little traps and we've succeeded at washing them and reusing them, and that reduces the cost. So if anybody's considering any of these practical aspects of control, that at least that is there. So it seems trivial, but in reality, like when we do outreach events, what like the stakeholders want to know is not some of the higher end things. They just want to know the practical things. So figure out some of that so it doesn't disappear into thin air.
So are there reviews on the subject, published review?
There is 2016 Peter Newman put one out, sort of like a broad look at the small hive beetle.
So is there anything in your review which is not there in that review?
Yeah,
what are the things that you have added not there in the review article?
There's the local concerns, I guess, the local experiences that we've had with the beetle here in Hawaii in this climate. And I can't remember off the top of my head, but there was a couple other things that I found in other papers that he didn't have in his.
And then are there any summary table or figure in the review?
Yeah.
How many figures and tables you have there?
I think I have. Let me check real quick.
Summarize something in the summary form in the table.
Yeah, so that's one of the other things I added too, is an updated table of the current prevalence, because since his paper came out in 2016, it's spread to like Korea, for example.
Okay.
Mm-hmm.
No, it has moved since then. So there are new areas where it's been found.
Mm-hmm. Yeah.
Why I'm asking the question, because sometimes if you write a decent review and it has new information and it may be publishable somewhere, if not in a very high-end journal, but at least somewhere. And minimum is a book chapter you can publish somewhere. or in a proceeding or somewhere you can publish. But one should target for a little better. So that's why it is always important to write the review of literature chapter on a subject very different from anything else in the past.
Yeah. Yeah.
Okay.
Let's see.
But your comment about about recent reviews, it might be worth to add like a little paragraph that just highlights this being a peer review published article, hit the previous one and reference that and say there are new contributions or differences, just highlighting it if somebody wants to go to the public source literature, but also what is different of what is new. And the small-eyed beetle in Hawaii has never really conformed to what was expected. So it's always had higher numbers. It peaks at different times than in the mainland. So there's all sorts of things.
These are the points then. These are the points. And then at the very first paragraph, you may say what is found in this review and what is already there. Yeah. Because this is a general way of writing reviews.
Yeah, because I think it would be good to even have like a table and but just put in first and front that there's this big review that was done, and then these are the differences we found, and that's I think a good idea.
So that will be done then. Then what is the next chapter?
So the next chapter, chapter 2, is about the small-eyed beetle and its relationship with the deformed wing virus. So that one, we're currently trying to get it published.
Well, what happened? Interviewing here. What happened was that one of the authors needed to approve it once they got to the journal, but he disappeared into the Amazon. So we were asked to basically hold on the submission until he comes back. So he went from the Amazons to South Africa. Thank you so much. So he's just flying to England this week. So we need his approval before the submission. So what Jason has right now in the chapter is more like a chapter regular version. So I realized he didn't pass on to you guys like some of the visual abstracts and more completed things that we did for the submission. So that's what I was saying. No, I think we have the more updated, but everything gets kind of frozen in limbo because of the automatic approval of people. And
and submitted to? To which journal was it submitted?
Well, why am I blanking out? Microbiology.
Microbiology.
Yeah. No, well, the
British Journal, microbiology.
No, not microbiology, but what is it? The same one Jessica submitted to.
Invertebrate pathology.
Yeah, thank you. So, but what happened was that it got frozen in limbo thanks to.
Okay, so it has this is a major chapter. This is a major chapter because you have everything, introduction, materials and method, results and discussion, everything.
It's laid out like that.
It is a major chapter. So, okay.
So I can just tell you guys what I have in that chapter, just lay it out for you guys real quick. So basically, there's a number of things that I did experimentally in it. So I guess to start with, we did a survey of apiaries in Hawaii. We went to 17 different locations we went to Maui, we went to the Big Island, we went to Oahu, and we went to Molokai. And out of those 17 apiaries, only 2 of them had beetles that tested positive for deforming viruses, Hilo and Pearl City. And in addition to that, at our Waimanalo apiary, over a period of 18 months, I took a sample of beetles every month, and then we ran all of those. And only 4 out of those 18 months had, or I guess the beetles tested to be positive. And then we did, then I did a feeding study. So the feeding study was a 3-part study. It started with a full life cycle rearing. I got some beetles from the field, and I had them lay eggs and raise them to adulthood, including pupation, on one of two diets, either bee bread, which is the pollen, and bee pupae. And so I tested them at I tested the adults, I tested the larvae, and then I tested after pupation, I tested the adults that emerged from there. And from all of that, the only ones that came out positive were the larvae that had fed on bee pupae. And then from there, I did. I wanted to investigate the transience, I guess, of the deforming virus in the beetles. So I did a couple diet rotation experiments. I did one with adults, where I got a bunch of adult beetles, and then I had them on a diet of either bee bread, bee pupae, or papaya. And then after 2 days, I switched their diets. So I had tested the bee pupae and the bee bread to be positive. I had tested the papaya to be negative. And so I rotated their diets. If they were on a positive source, I switched them to papaya. If they were on papaya, I switched them to bee pupae. And then I waited a couple more days, and then I tested them. And out of all of that, the only, actually, for the adult ones, none of them came out positive. There was no positive adults at any point during the adult diet rotation. So then, being that in the first part, we did have positive larvae that fed on the pupae, I did a similar diet rotation experiment with the larvae. So I got a bunch of beetle larvae. I had them on papaya first, which I had tested to be negative. And then after a couple of days, I switched them to bee pupae, which I had tested to be positive. And then from there, I waited a couple of days and switched them back to papaya. So what I was trying to do with that was I was trying to see if I could induce a positive result in the larvae by getting them on the bee pupae diet, and then see if I could show that it clears, or that it was just like a temporary positive test, like the infection doesn't stick, or like and so the results of that was, so I tested them at all three stages. At the papaya stage, they were negative. After the bee pupae, they did test positive, and then after I switched them back to papaya, though, they tested negative. And then I also tested for the negative strand of RNA to look for evidence of replication, and it did show a positive. I did an increased replicate. I guess I tested 12 individuals of larvae, and then and it all came out positive.
Okay.
Mm-hmm.
So the virus is replicating in the beetle.
Well, I was thinking, being that after they switched to papaya, they were all negative, that maybe it could possibly be due to gut contamination, like whatever they have eaten. If the virus is still, I guess,
tissues.
Being that when I tested the bee pupae, I mean, they were positive the same way.
Okay, this is, I think, Spokane is super hard, and I was like desperately trying to find. So there are three diets, right? So the store pollen was positive, the bee pupae was positive, the papaya is negative. And what Jason did, and maybe he can throw it, was derived from the fact that there was a previous paper, the only one that has looked at deforming virus in the small hive beetle, and it did show positive. So that paper showed it as replicating in the beetle. That paper showed it as a potential reservoir of the deforming virus. The problem is Jason's preliminary data with all the 18 months that he mentioned showed very low levels of very low prevalence. So if the thing actually replicates in the beetle, if you went out in the field and collected lots of beetles and did it over a long period, you will think that you will have more hits, more positives. So that's how we follow that direction, was the field data that we're catching is not matching the lab work that was done with the beetle. And so then we tested different products. And what Jason found was that likely to be found on the larva more than on the adults, even on the lab, but it was like really not happening in the lab for us. So, and the other thing is that Leslie said that it was transient. So if the larva had it in it, it didn't go. When it pupated, the adult that derived from that pool of positive larvae were all negative. So
we
that
did
was
the
the
single strand RNA stuff. When was that positive?
The negative strand. So it was positive in pretty much all of the ones that tested positive, I believe.
In a people stage, it was some of them are positive, but later on in adult stage, it became negative.
Yeah.
How do you explain this? Did you discuss that in a discussion? Why it became?
So
give a proper explanation for that.
Yeah.
What is your explanation?
That's your feeding experiments, right?
From larvae to pupae to adults. So that one, I mean, I was thinking it could be the transients of the infection in the same way that during the larval rotation that they were positive, and then after a couple of days of eating papaya, they became negative. I mean, that's probably the main one I would think, but it also could be in that one from the pupation. It could be due to the physiological changes that the beetle undergoes during.
Is it possible? Suppose let us say you have a 100 pupa, 100 pupa, and then you take a sample of that, they got positive, but then when they grew to say adult, so they were uninfected in this population, they become adult. Is it possible?
If they were
so originally it is a mixer population. Some of them are infected, suppose. So now when you take a sample here, you found this is a sampling error, so you got the positive one. But then later on, when they develop into adult, suppose the uninfected one become adult. Is it possible?
Yeah. I mean, I guess possible.
But these are lab experiments, right? So are you sampling every single individual that you fed?
So that's for the first one where I put them in the dirt, like upstairs on the roof. I had them in little containers, and I reared them from larvae to pupae to adulthood. But yeah, I mean, I guess there always is that possibility that maybe, I guess.
Well, the whole
I can't say for certainty, but
Jessica, work that is coming after Jason, is that there's more and more problems in the literature with if you get a positive result for replication, the problem that people are encountering is whether it's a gut contamination, so is it food that is going through your belly that gives not only presence, but like the semblance of replication. So in this case, if the pupa is a bee pupa, and bee pupa actively replicate the virus. So if you eat that, is it possible that you will show yes, and so that is a problem with the interpretation that more and more people are asking. That is, the firmware has gone through people asking, does this have the firmware? It's just positive. Then it's been, what type of the firmware is? Then it's been like, is it replicating? And now people are saying, oh, it has to be, is it replicating in the tissues of the animal? Which then it means more of an immunological process of determining, what proteins does that particular species secrete, for fighting pathogens in their system. So people more and more want a proof that is sexual replication in the tissues, not inside the gut. And it's not always practical, obviously, to try to dissect guts and do it accurately enough that you don't get contamination. So the best thing would be to find markers that will tell you that it's in your tissues. So in a sense, the paper that was published with the small hive beetle wasn't that long ago, and that was saying, oh yes, it's going to be a reservoir, it's going to be the new thing, blah blah blah. But then when we were doing that sampling with Jason, they were just not showing up as having it.
Yes.
And the ones that showed positive in the field had to do with slime outs, with when there's more food, probably more mushed up larvae that were attacked by varroa all over. So the field data really helped to try to track something that was done in the lab and that sat there for a few years, like untouchable, because it came from a very good lab and nobody kind of wanted to mess with that. So when Jason's paper gets published, I think it would be a pretty important paper because it will be arguing something that is now very hot and is you can't quite assure that replication is really happening. And I mean, his reversal, like you said, from larva to from positive to negative just by switching diet in a couple of days, that is pretty telling. The field data is pretty telling. So we're going, I think, in the right track. We still don't have any results. We haven't done any, labeling, tracking proteins for that response. But the data that he's getting points in that direction.
Did you discuss these things like that in the paper?
I mentioned some of them, but I still gotta clean it up. Still gotta flesh it out. The main practical implication that I'm thinking about getting at with this part is that it seems like the beetles aren't likely to be like a very important, source of deformed wing virus for the bees, like at least not in the way that varroa
which is very important here in Hawaii because the beetle is all over, even in places where there's no varroa. So places where there's no actual vector for varroa, like Maui, Molokai, Lanai, all those places, Kauai, don't have the mite. But if you go by that paper, we'll be more in danger from having an insect that could carry the virus. So with Jason, again, his relevance can be very local, even though the meat of the problem is very more international, more global, more current, a bigger topic. So, but yeah, the practicalities, and I was a pain to review those things.
Mm-hmm. So, since this paper will have a lot of interpretation. So interpretation, people look at the materials and method very closely. Did you write a good materials and method describing the experimental?
I tried to, I tried to.
But Jason, what we can do is you can send the newest version before the latest changes from Martin come in, and we get the submission, reroute it, and then you guys can look at where everything is, including the visual abstract, which clarifies all that, because we struggle so much. We had this, you showed me the previous version and we were like, oh God, you can't follow this unless you're like been there. So
I will not be able to catch problem here, but I am just, I am telling you where people look at it, because the paper, the interpretation is complicated, because the interpretation can be done a different way. Mm-hmm. So one has to look at the materials and method closely. So people will look at the materials and method, especially other entomologists or virologists. Not me, I do not know how to look at it. How many figures and tables do you have in this paper?
So this one, I believe there's probably around 4.
4 figures?
Yeah.
And table?
Including figures and tables.
Okay. This one, I did provide comments on.
Yeah, you did. Yeah, that was really helpful. Thanks, Will.
And then, but there's a revised version now.
It's undergone a lot of changes since then.
You are planning to write the thesis chapter as independent, like a manuscript, huh? So you will have introduction, materials and method, results, and discussion.
Yeah.
And then also literature references at the end.
The works cited at the end. Yeah.
So similarly, the first chapter that you will like review of disaster, that will also have the similar style.
Yeah.
More or less, although
Yeah.
there will be no materials and method
Yeah.
there. Materials and method or results there.
Yeah.
So you have now completed 2 chapters.
Yeah. I mean, I wouldn't say completed, but they're coming along. I feel like they're pretty good shape. Okay. So then chapter 3 is about the in-hive distribution of the small hive beetle, where they hang out within the hives. So, so in 2016, I did, I guess, 16 observations over the course of 3 dates, in which we would lift the beehive off of the bottom board. We would count
Explain
how many.
first why.
Oh, right. So previous literature has suggested that most small hive beetles, they congregate more on the bottom of the hive, and then the importance of this is that when most of the time people use traps to control the beetles, and then when you're using traps, the location of the traps is important. You want the trap to be where the target is and not where the target is not. So yeah, so previous literature has suggested they hang out on the bottom, but from what we've been doing working in the field, we've noticed that most of them.
Why does that matter if they hang out in the bottom?
If you don't have the trap in the right place, you won't be getting optimum catch of the target. So the point of this experiment was to investigate where the beetles like to congregate the most within the hive for the purpose of optimum trap placement within the hive and type of trap, because there's different styles of trap. There's traps that go on the bottom board as a replacement bottom board, and then there's the ones that we use, which are between frame traps. They go between the frames, and we put ours where we've noticed most beetles, on the highest part of the hive, the top box, whatever it is, and on the sides. So to investigate this, to test this, we lifted the hives. The reason that I was thinking that the previous literature reported or suggested that most of the beetles were hanging on the bottom is because of negative phototaxis. We were thinking that they found most of their beetles on the bottom because when you open the hive, sunlight is coming in from the top. The beetles run away, so they run to the bottom. So to investigate that, we started by lifting the entire hive off of the bottom just to see how much there was present in the bottom first, and then we put it back down, and then we opened the hive from the top, and then we counted the regular way. We checked the end frames, we checked the middle frames, we recorded the numbers of beetles that we saw, and we worked our way down until we got to the bottom again. And so I compared hives that had no super boxes, so a hive that's just made of a single box. I compared those against each other, and then I also tested some hives that had one super, so a box or a hive that's made of 2 boxes. And I used a one-way repeated measures ANOVA with a Tukey multiple comparison, because I did survey some of the same hives. I know, Mark, you mentioned the factorial analysis possibility, but that was the reason, or at least that's what I'm thinking. And I used one-way repeated measures ANOVA. Yeah. And then so the findings from that was for the hives that are made of 2 boxes, 1 super box, there was significantly more beetles found on the top sides, just like we thought, compared to any other location in the hive. For the hives that didn't have 1 super, for the ones that had it was just a single box, there was a higher mean number on the sides and the bottom board, but it wasn't significant. The P was 0.057. So I mean, it was It was close, but like you mentioned, Mark, there's like a 20% chance, so like a 1 in 20 chance that it
So please tell me
that
again
it's
the difference
wrong,
between the two.
that's
The
insignificant.
first one you keep hive over hive.
Yeah, two box hive.
So then
yeah.
they stay in the top mostly. Yeah?
Top sides,
Top
yeah.
side.
Uh-huh.
But if you keep in the only one hive,
Yeah.
then they go to the bottom because of the light. Again, supporting your hypothesis.
Yeah. The problem with drawing conclusions from that one is that it wasn't the P was a little bit higher than 0.05. It was 0.057.
Oh, what do you make of it? Have you been looking at the things that people are saying about P values these days?
I came across another paper. It was actually another small hive beetle paper where the author was arguing that even though numerically it's statistically insignificant, I think the way that she worded it was,
it's an arbitrary thing, right?
Yeah. The way that she worded it was for a beekeeper and for practical purposes, even something that's slightly marginally insignificant could I guess be useful for a beekeeper in a practical sense. But then
P-value is only useful to you. Honestly, anyone reading that is evaluating your mean. I mean, so
yeah, and then but then if I include that, I gotta argue that
So how do you explain that when you have two boxes, it doesn't get the light or what?
So I mean the general trend seems to be the same. Like there's less beetles on the bottom than the sides. It's just that with the single box hive, the p-value was a little bit higher than 0.05, so it's technically not significant at that confidence
But there's
interval.
big differences between the first look at the bottom board before you do anything to the hive to the second look. And if you're a beekeeper, you're working your way down. So what the beekeepers tend to observe is all the beetles in the bottom. And the assumption has been that that's where they hang out. And I don't know why, like it doesn't make any sense for me, but people think, oh, it's because the top, there's more light. Well, 99.9% of the time, they're equally dark because everything is closed. But everybody just for some reason assume the darkest spot is on the bottom board because it never gets light. And it's like, it's very strange that the light. Yeah, no, but I'm saying that when you talk to people, they say, oh, the darkest spot, of course, further away from the light is the bottom, but it's as if the colonies had like a transparent lid the whole time or something, because, it doesn't, once you close it, everything becomes dark in there. So, and I think the hardest thing for people, and that Jason needs to do a good job at explaining, is that the control of the beetle is like a fine balance between the population of the bees that are going to chase them around and the space you give them. So if before, even with varroa, people could stack big boxes of honey supers on top and let them be. And just if they were empty, they were empty. If they got filled, you could take them home. But as the beetle got there, any place that has a little bit of food is a potential problem if it's unguarded. So what I think Jason needs to really make sure of that is that this challenges, especially tropical beekeepers that are not used to taking away supers that you can leave them there all year long, to incorporate a different step for management that can change really, really fast with your colony.
So in wild bee hives in Africa, where the small hive beetle originates, what's the distribution in those colonies?
Yeah.
In the Pharaoh colonies?
Yeah, wild native African honeybee colonies in Africa that live in trees or holes that are often exposed to light where the beetles find those things.
I don't know if anybody's specifically investigated. I remember seeing a figure in that in the 2016 paper where
the Newman.
Yeah, he had an illustration of a wild hive, and he had
problems with it.
I mean, he showed some beetles on the bottom, but he didn't specifically address
So like sitting from the bottom of the comb.
yeah, or like in a hollowed out like tree opening, like below. He had them pictured below the comb as well as some on the comb, but he didn't really address that in his paper. Like he didn't really comment about it.
He had something like this where he had like beetles saying that beetles could easily reproduce in the bottom, and beetles in different areas of the comb. The two things. One is that in managed bees, this will get cleaned up. Beekeepers go in and out and routinely clean the bottom. In a feral hive, there's likely to be more crud that accumulates in the bottom. And if the entrance is too high, the bees are not going to spend energy going up and carrying things. They're just going to leave it to rot in the bottom. So the beetle could potentially.
So a lot of them don't occur in cavities like that. They're just like exposed completely. Walk up to the combs. So what about that? Do they consider hide away in the comb and reproduce? Or are those exempt from damage?
In those cases, I'm not entirely sure. Yeah, I was under the assumption that most of the honeybees are. I mean, I guess maybe the European ones. They usually try to nest in a more protected area, and that it would be rare to find them like just on a branch with comb exposed to like the rain and the sun. Yeah. Oh, I also to further back this up, I on 2 more days I checked 23 hives, and so we checked the bottom boards first. We lifted the whole hive except for the bottom board, and then we put it back down. We left the lids open for 5 minutes to simulate a beekeeper going through it, and then we checked the bottom board again, and then there was a significantly higher number of beetle found on the bottom after leaving it open for that 5 minutes compared to initially.
So that is because of light then?
It seems like it, yeah.
And it really does, and again, more stuff needs to be included regarding the management. But people you probably know, people in Hilo and very wet places had a big problem with beetles, and they opted for a bottom board that is just a big mesh, and they were putting like huge tubs of oil underneath, and they were catching lots of beetles. But again, they usually were catching lots of beetles because they were doing, working the hive, and everything was like scurrying, and then they would fall in these oil pits. But the problem with that management, it depends on how frequently you check the hive. And also, if you put some things where it's this deep full of oil, you need a hive, that tub needs to slide in and out, and you need a hive that is very level, because otherwise, if you're trying to pull it like this, you're going to be splashing oil all over, and that's going to kill bees. And a lot of the times, ground is never level, and second, we try to aim the colonies down so water like scoots out, so when you're pulling, it's like a wave of oil. So for us, we thought it was incredibly impractical, and this is the reason that I'm encouraging Jason to not to criticize that method, but to say that with a much smaller little container of oil on the top, if you believe the fact that they actually live, are chased to the farthest away parts of the colony, that you can do just as well rather than having that very cumbersome monster oil mess in the bottom.
So what do you get when you put the oil on the top?
When you
put the oil on the top, as you said.
Mess of the traps, yeah.
Yeah, you. We catch beetles in them.
They are equal number or good number?
Yeah, usually a pretty good number.
What's the average?
The average, there's a lot of variability between hives. Like, it could be anywhere from 5 to 100
per week. Yeah.
Yeah.
So, how is your writing going on this chapter?
So this one, it's going all right. I need to, I still need to clean it up. I still gotta flesh it out. With this chapter, and then the next chapter that I'm going to talk about, this discussion is something that I need to work on a lot, and I was wondering
this chapter have you written or not?
I've written a draft of it.
Okay, you wrote a draft. So
Yeah.
You wrote the results. Huh?
Yeah.
Results. Yeah.
The results I need to make it nicer also.
Okay.
Yeah.
When you write the results,
Uh-huh.
the headline of the results, do you first write it? First the headline before writing the details. Write the headlines. Headings of the results.
The way I have it with results as the heading, and then I have the figure.
Results has several headings. Huh?
Subheadings.
Oh yeah.
So those headings you discussed with Ethel and figured it out.
Yeah. Yeah, I got to put subheadings in. Mm-hmm.
Yeah, but there's some data, there's some information that I'm trying to pass to Jason because it comes from Latin American countries where the USDA has made some really lame suggestions, like the bottom board, because a lot of people don't have that mesh and don't want that oil. They suggested that they drill a little hole and put the equivalent of a Coke bottle at the center of the bottom board with oil underneath and like either paint it black or cover it in tape. But again, number one, you probably put it in the wrong place and you have an opening this big. That would be, so it has to be the unfortunate beetle that just happened to be chased past that hole that is going to, fall. But these are the suggestions that I think are made, even international suggestions that are made based on poor observation or lack of follow-up on something. Very, very simple.
I think when I first saw this chapter, I was concerned about the lack of depth.
Yeah.
Very superficial. Yeah. And this is a PhD, right? You're supposed to explore things that are of bigger implication and so on. And give a more sort of synthetic perspective on that issue. Yeah. So you see yourself being able to do that with this chapter now, for example. I can see the previous one. But how do you make this addressing the bigger picture? Contributing.
Well, I mean, I definitely think that it does have a lot of practical importance for beekeepers and significance, but yeah, I have been struggling in this chapter and the next chapter that I'm going to talk about with I guess things to I guess cover, things to touch on, and I can tell you guys about my other chapter first, but yeah, I was wondering if you guys have anything off the top of your head, just like, I mean, not even thinking too deeply, like things that would be good to touch on, I guess, in the discussion.
Your thesis, your chapter will have depth provided. You can put into war all the things Ethel and you have been describing in terms of interpretation. Why there are more beetle in the side? Why some other experiment? One probable reason for having so many beetles in the bottom, and how oil affected the presence of the beetle population there. The interpretation, if you write, that will bring some depth to your chapter.
Yeah.
If you can really good in writing and putting all this interpretation in proper place.
Yeah.
And how many figures and tables again?
This one, let's see. I think I have about 3 figures in it.
Do you make the figures and table ready before writing chapter?
I kind of, actually, yeah. I when I was doing the analysis, I made the figure to visually help compare. And
then do you have more figures to make?
For this one, I was thinking about including a sort of a similar figure that I'm going to include in the chapter 1, just a picture of the overall layout of the hive.
Should it be a picture or it should be a sketch?
Well, Jonathan Wright's been helping out a lot with he has some really good computer art made pictures
Sometimes
that he
can
let me use
sketch,
for some of
yeah,
those.
like this, hand drawn
Yeah, like hand sketch.
or
Yeah.
Yeah, thanks to
Uh-huh.
John, we have like hives that, so they look line drawings, but
line
very
drawing,
accurate
line drawing,
3D.
yeah.
And you can see, so you could put like the percent that is on
Yeah.
the lid, the percent that is on the super, the percent that is here, the percent that is on the bottom, because you have them like floating in space slightly, you know, in the right order but separated. So Jason has those, and they look really nice, and I think they will be good visual aids. Let's just
I
say.
think that can be in your Michelson method. Yeah? Figures
Mhm.
can be there in the Michelson method also.
Yeah.
Because you see, it is very difficult to describe this kind of experiments without proper
Yeah.
figure what you are talking about.
Mhm.
No, it
Yeah.
is very hard to imagine if you're not a beekeeper. 'Cause it becomes like, I don't know how people, well, like, you know, like reading to become a beekeeper is not a good thing. It's very visual, and you've got to imagine the 3D space.
The first chapter, as you described, the second chapter which you described, the previous chapter you described,
Yeah.
that has I see some depth now coming into that. Yeah,
Uh-huh.
that one. This chapter, if you can incorporate this discussion, it will bring into that. Otherwise, it will be kind of this question that Mark is raising, and which I also seconded earlier.
Yeah.
That will come. Yeah. So you need to give in emphasis whatever result you get, the interpretation and possible interpretation, with the supporting the citation. So
Mhm.
somebody got, and but this
Yeah.
that you need to bring it to this.
Mhm.
And when you make your presentation, this should be in your top of your mind so that you can
Mhm.
explain fluently.
Yeah. Uh-huh.
You
Yeah.
need to practice your presentation.
Yeah.
Yeah. So many times.
Yeah.
So actually many times
Mhm.
presentation, even to talk with us,
Mhm.
you need to really practice and prepare
Yeah.
well, yeah? So that you can, if we ask one question, you can elaborate.
Mhm.
Well.
Yeah.
So one suggestion for the discussion or
Yeah.
something to discuss would be the role of negative phototaxis broadly, and like what significance does that play in the species? Like going back to what Mark was talking about in its native range, like why does it have this behavior? What would be the normal, wild type situation?
That's actually really interesting, because people who have kept, you know how you have observation hives like in places, with a glass cover. The beetles don't like it as much, even though they can totally survive. And those hives that have more light. Not that you could do anything in the field, but there is something very strong for the beetles. Because one of our concerns, and we haven't gone there, is that we wanted to make some for organizations in which you would have this, but we were afraid of the beetles, creating a slime map, because it's, you can't manipulate those hives inside the glass thingies as quickly or respond as well. But the number of beetles that hang out in those hives is lower, and they're unhappy when they have more light. Yeah, they just don't. I mean, and those usually.
Why would you not do that?
Huh?
Why could you not do that? Like plexiglass hives.
Well, we were concerned about.
Okay. I mean, as far as beekeeping, have you mean like transparent hives? I would think the temperature would be a concern. You have direct sunlight coming in. Even with the shaded top, I think the bees' optimal conditions are more dark than having light coming in through the side.
Glass is not very insulating.
Yeah. Uh-huh.
So how do bee keeps it dark when they make hives on the tree?
Well, from my understanding, most of the time when they nest in trees, they often don't make their hives exposed. Like they gather like on trees to swarm and stuff. But when they actually start building, they usually choose somewhere more like inside of a tree, like a cavity. And then that's why in a more urban setting, you always hear about people having like I've got bees coming out of my roof. I got bees coming out of my ceiling. They're in the walls. They're in sheds.
They can, but usually when you have something very exposed, it's this protection against rain in somewhat shade. But I think the interesting part about them, though, is as Jason knows, and this is the same inside a regular hive, is your brood and your queen and your most activity will be here. So that part is actually pretty shaded, and the outside things are honey. So your core activity is more shaded than the thing. But they can go down with beetles too. So
you need to talk with Ethel more frequently to pick up the vocabulary, because when she's talking, she's using certain words you are not using. For example, she's talking about core activity. Your vocabulary must improve, and that will improve when you talk with her more frequently.
Yeah.
Okay. And then, kind of related to that, like, are there ways that you could use these results to your advantage? So, is it beneficial to open up the hive frequently and drive them to the bottom? I mean, could you place your traps at the bottom, even if that's not where they're normally hanging out? Like, you're finding that open them up, or driving them down there.
LEDs in the roof.
Yeah.
How about having LEDs in the roof? Like
solar.
Yeah. Oh yeah. Yeah.
How do we
get LEDs?
Yeah, exactly.
Solar panel.
I wonder if you could put LEDs inside and just turn them on and see if they go down. That would be awesome.
How to remember what people are advising or suggesting?
I'm writing it down.
You're writing it down?
And I'm also recording it. Yeah.
Oh, okay.
This predates Jason by a long time, but one of the first presentations I saw from Hilo, when the beetle came, there was a talk that said, don't open, and actually those were the recommendations from the mainland, don't open the hives because it attracts more beetles. So it was saying that the pheromone of alarm of the bees was attracting the beetles and you should not mess with it, which would, in reality, would be absolutely counterproductive because you need to know how big they are to adjust the space. But one of the first things that came out was don't open your hives. And I think it was, and then somebody in Hilo opened a hive that hadn't been open and had like 300 beetles, and they were using it as a example, because they were at the bottom. But I'm like, you just had like a major beetle party and it just went down. But it was, they were very unhappy with my comments. But it was, over time, it was very obvious that, but again, like I said, the beetle hasn't conformed. We have slowly learned to almost like go against the flow. So I feel bad for these guys because, I was not a social bee person, and we had to like say no to big wigs and say, not what we're seeing. So it's been like a learning curve of becoming more independent, and certainly things like don't open the hives, or I actually don't have the time, but I am convinced that whole thing with the smell of the beetle was a mistake, because I think they attracted to anything that smelled bees. And I think they put a beetle in a Y tube, and they put a bee in one, and the bee was freaking out. And of course it was, put in like an alarm pheromone, and the beetle went to it. And then from there on, which I think is an incredibly stupid experiment, from then on it was written in the literature, and it's been there for like 15 years, that the beetles are attracted to alarm pheromones. And it just keeps being quoted and quoted. And if it were because of that, and we open our hives like sometimes 2-3 times a week, we would be the ones up to our ears in beetles, and we're not. So, but to refute that would take a lot of effort that I have no interest in doing. But I just think is really lame. So the small hive beetle wasn't a hot topic for a long time until it's found in Europe, and then it became a hot topic again because the Europeans are worried.
So what else is there, or is there anything to add in this chapter? Is there anything, or you finished talking everything?
In this chapter, yeah. Yeah.
So far you have described 3 chapters, 2 chapters of results and the introduction.
Yeah.
3 chapters.
Yeah.
And what is the 4th chapter? You say 4th chapter.
Yeah. So the 4th chapter, it previously was 2 different chapters, chapter 4 and 5. but I'm thinking now that it might be best to combine them into just one chapter 4, dealing with the population dynamics of the small hive beetle. So, so the 2 parts that I combined was management related issues and seasonality. So for the first part, we monitored our apiary beehives from 2013 to 2016. We would go in every week. We would count how many beetles are in the trap. We would make note of any important hive events, like if there was a queen loss or something like that. And for the management part, first I investigated the effects of honey harvests and formic acid treatments on small hive beetle levels. Those are 2 procedures that beekeepers often subject their hives to,
and that have been have been named, like opening the hives as potential disturbers.
So honey harvest and what?
And formic acid treatment for varroa mites.
What happened? Formic acid, what does it do?
It's to treat for varroa mites. Yeah.
It's like an internal fumigation.
Yeah, it's a patty that you put inside your hive, and it's the volatiles. It releases vapors.
Formic acid is volatile.
The form that we use in the hives. Yeah, the vapors will permeate through the cells and kill varroa mites.
It's a slow release. Yeah, so it gets the patty gets drier and drier as the formic evaporates. So supposedly it doesn't hit him as bad as a flash treatment.
Sorry for interrupting one thing. The question of death phenotypes will come again and again.
Yeah.
So one way to handle this, and to address this, is to everywhere you have to think about writing the problem very well. What is the problem? Why you are going to do at the introduction?
Yeah.
So you have to write it, describe the problem.
Yeah.
I don't know how you describe, because that will be pretty important. And then if possible, you have to then you bring some kind of important literature, few things, not entire literature, and then based on that you try to form a hypothesis.
Yeah.
You see that? And then then present your results. Then at the end, you support or reject the hypothesis. Then only it will bring kind of a more depth to your thesis.
Yeah.
You see that?
Yeah.
So the bringing suddenly, before telling the story, you have to tell in such a way the problem, why you want to do. And then you bring other people finding little bit of ideas or general idea. Yeah. And then based on this idea, you cannot have a hypothesis suddenly from nowhere. It has to be based on some kind of a previous results or ideas. Yeah. And then you hypothesize. Yeah. And then bring importance to your work. Yeah. And then describe that one. That will be kind of a challenging. PhD work. Yeah. You understood what I mean?
Yeah, I understand. Yeah. Okay. Yeah. So the overarching.
You see, so far I have not heard anywhere hypothesis work.
Yeah. Okay.
Somewhere you have to bring the word hypothesis, because generally a PhD research should be hypothesis derived.
Yeah. Yeah. Okay. Yeah. So thanks. So so going off of that, the overarching theme of chapter 4 is the problem of small hive beetle populations. I wanted to investigate what could possibly be associated with an increase. So looking at management procedures is the first part, honey harvest, formic acid treatments, and then also investigating seasonality, rainfall, temperature, certain aspects about the colony phenotypes, like how much bees the colony has, how healthy it is, any important queen events. So these are all things that I wanted to look at in regards to small hive beetle population increases. So for the first part, from what we've been seeing in the field, I hypothesized that honey harvesting and formic acid treatments wouldn't have a significant effect on small hive beetle levels. And so to investigate that, I recorded the dates that we did honey harvest. I recorded the dates that we did formic acid, and then from 2013 to 2016, every week we counted the number of beetles that we had, and then so I used the t-test to compare these. I tested a period of 2 weeks before harvest compared to 2 weeks after the harvest.
2 week what?
Huh?
What are you comparing?
Oh, the number of beetles found, in the traps. And so I did a paired t-test for every hive that underwent a harvest, that period of 2 weeks before and then period of 2 weeks after. And then there was no significant difference in the number of beetles that was found before the harvest compared to the period 2 weeks after the harvest. And then for the formic acid, on the other hand, I ran it in a similar way, 2 weeks before harvest and 2 weeks after harvest. But not harvest. 2 weeks before treatment, before formic acid treatment, and 2 weeks after formic acid treatment. And then in this case, there was actually a difference. There was and it was significant. There was more beetles post-formic compared to pre-formic treatments caught in the traps. And then
do you have a control for that, or do you have hives that weren't treated before and after checks?
I do. I could throw those in and compare those also against everything.
Because what you would want to do is
include them in your data set.
Oh, include them in my data set. Yeah.
But I mean, what you would be looking for there would be
significant differences between
one and no change in the other.
Yeah, like if your controls are not changing, but because if it just happened to coincide with like seasonal.
Yeah, that's a really good point. Thanks. Yeah, I'll do that.
So getting back to the harvest thing.
Yeah.
I would think that small hive beetle would be less likely to be in the super than in the brood box, right? So why would harvesting impact them at all, other than lights driving them down? Or is that not correct?
It's from the previous the experiment, we found that most of them usually are, or more they are more often in the super. But in regards to in regards to the reasons that they might be impacted by the act of honey harvesting, it is a disturbance to the hive. The way the harvest works is that the beekeeper has to take the lid off. They lift the box off, the whole box, and they have to shake out the bees. And in our case, the beekeepers we work with, they have these big backpack leaf blowers. They put the hive on the honey box, the honey super on top of the hive, and blow the bees out while also brushing them out with a brush.
And they're gonna take the supers.
Yeah, and then the supers are taken off to be harvested, and then brought back and put back on the hive. So it is
when they brought back, what do they look like, and what happens to the bees?
Mm-hmm. What did the bees look like?
No, the honey supers.
Oh, the honey supers. Yeah, when they're brought back, they're devoid of honey for the most part. I mean, there's some traces of honey that didn't get extracted. It's mostly open honey cells, wax and foundation in the boxes.
What do the bees do?
And the bees will rebuild and start filling them up again after they're placed back on the hive.
There is a big change in activity when you bring what is called the wet supers, the empty supers for the bees to finish cleaning up and rebuilding.
Yeah.
And some
And
of
it
the
is a big
local
physical
beekeepers
system.
complained that they lost hives to small hive beetle. And it's usually, they'll usually complain that it was my strongest hive. It had tons of supers. It was great. And then I brought it and then it collapsed like 2 weeks later. So,
Well, that could be because they were hanging out in the super and then rushed down into the hive at harvest, right? Could that be? And then they impacted the brood.
It could be like if they're hanging out in the honey super and then once harvest happened they go down.
Unless you open it, yeah.
Yeah, I mean, yeah, they probably do run downwards into there.
And where are your traps for measuring the numbers?
The traps are in whatever is the highest box, so the top of the hive on the end frames where we found most beetles to congregate from the previous.
So the end frames of the super.
Yeah. Yeah.
but if you decide, for example, when you come back that the hive, that there's no flow and that they don't need a super right away, that also means you're going to put the traps in the deep. So one thing that just talking to you guys occurred to me that you should look at is if the configuration changed. Because if it is not just simply before and after, but before and after, which with a reduction of space, a reduction of space may force more, it may facilitate or hinder whatever, I don't know, the capture if you remove a box, So it might be worth going through your data real quick and selecting those that after the harvest get shrunk, basically, at less volume.
How many hives total have you been monitoring small hive beetle with?
Around 30.
For that period, to 2014 to 2016.
Yeah. Yeah. Numbers would fluctuate a little with like colony losses and colony additions.
Yeah, sometimes there are as many as 37, sometimes like lowest was like 29, but usually very close to 30.
So have you analyzed and organized the results for this chapter?
So I mean, not in depth. Yeah.
So you have not made the figures and table yet.
I have some figures, but I'm gonna do what Will was suggesting and add in the.
All right.
See if I can.
So you first make the figures and tables
data.
and see what is missing to complete this chapter. How do you see what is missing to complete this chapter? How do you find out?
By running it by Ethel and you guys. You guys are thinking about it critically.
It has to be a story, little more or less complete story. So to make the story, something is missing, and that you by discussing in detail, you can come out of it. And then you have to find that missing part of the data. What is missing? Yeah, you usually discuss and find out what is that. That is how we write the papers and we make the figures and tables first, and then we discuss. Oh, this will not be enough. This is not a complete story. So you do this, and show me, and then we will have a better story.
Yeah. Well, Tristan, how about the newest thing that we were discussing with the seasonality?
Oh, yeah, I'm getting to that. Yeah.
Okay.
So, is there anything more to discuss about this chapter?
Yeah. There's more to discuss. So that was the management part, honey harvest and formic acid. For the seasonality and the colony phenotypes. So I did a couple of things with this one. I investigated precipitation and temperature. I ran a linear correlation with beetle numbers against temperature and precipitation. Unfortunately, it was pretty messy, and there was nothing significant there. For 2016, we started recording more detailed data about the hives. We recorded the number of bees that were in the hive, and also the health of the brood pattern, and we did this on a numerical scale of 1 to 3. 3, as far as for bee numbers, for example, 3 meant that it was almost full of bees, whereas 1 implies like 25% coverage of the frames with bees, and 2 would be about 50% coverage of bees. For brood pattern, we also assigned it on a numerical scale of 1 to 3. 3 is the healthiest brood pattern. Brood pattern health, it's evaluated based on how spotty it looks. If it looks like Swiss cheese, if there's a lot of empty spaces and holes, it's generally indicative of poor health for the colony. So if it was a very unbroken pattern, if there was minimal holes in the brood pattern, we gave it a score of 3. If there was if it's like a very spotty brood pattern, it got a score of 1, and if it was in between 1 and a 3, we gave it a 2. And so we recorded that. We also recorded queen status of the hives. If there was any queening events, like if the hive went queenless or if it re-queened, I made note of that. And so I ran that in a linear mixed model. I used a linear mixed model because in another paper that I had read, they also used a linear mixed model to investigate similar aspects of that. And the results of that was that the only things that came out significant was, interestingly, the average bee number, so the average number of bees in the hive, and also temperature. So queen status came out insignificant, so did precipitation, so did the brood pattern, and so did size. Size was the other factor that I recorded, how many boxes the hive was composed of, and that was insignificant.
So what kind of model did you run?
Linear mixed model. So I ran it with hive as subject and dates as a repeated measure, because that seemed the most appropriate, because I did record the same hives over multiple dates. And
the response was number of beetles.
Yeah,
During that sampling event, or like, I guess.
Mm-hmm.
Okay.
So I did it. I put them together monthly. Yeah, monthly aggregates. Yeah, and then so also, or in addition to that, just from visually looking at the data, graphing out the dates and the population levels observed of small hive beetles in the apiary, it did look like there were spikes in population levels more during the winter times. And so I did compare the average number of beetles per hive for the periods of November, December, and January compared to the other 3-month periods of the year for that time period. And there was significantly more beetles during those months of November, December, and January. What we call the winter season for those years. And also, I was talking with Ethel the other day. We were looking at some more things in the data, and it looked like between the years of 2014 and 2015, during that winter period where we experienced that increase in beetles, in 2015, there were a lot more queen events going on. We were having a lot of queen failures, issues with the hive, and we did have, just glancing at the data, we did have a lot more beetles during that time compared to the same time period in 2014, where we didn't have all of those requeening issues happening. So we're planning on looking more in depth in that.
It also coincided with 2015 was a year that was completely crazy with like storms. So the rainfall was from like, I forget if it was like September, it was just like continuously way higher than ever predicted, I guess, and just straight through for months. So I think from the point of view of the bees, I think it affected their ability when the queens left to find a mate. I think it may have affected them. So Jason wasn't, although he had very good queen data, he wasn't really tracking like the survivorship of those queens. Like how long do they last? Do they come back and stay and do okay? But because I was helping Ning do something with the queens, I said, oh, I wonder what's in Jason's data. Then when I saw that and I looked at his data, then we started talking about the possibility. So the problem with running some of no one else has been able to find a correlation, which is really weird, between the phenotype of a colony and the number of beetles. So people have worked in South Africa where it's native, and even there they haven't been able to find an identified characteristic that goes with it. And but that peak, that very initial peak compared 2014, 2015, and even 2016 falls back to normal with the previous year, makes you wonder, how do you tease apart things where two organisms may be responding to the environment kind of at the same time by having very different effects? And first of all, the beetles here peak in the time where the colonies are at its lowest in Hawaii, which is really the opposite in the mainland. In the mainland, the highest peak number of the beetles happens in summer, when the colonies are the strongest. And here the peak when the colonies are the weakest. Why? We don't know, but that's our highest number. And it's not just capture, because you could say, oh, the colonies are small and you put in smaller boxes and you capture more. But no, you go through the colony, you poke at it, pry it, everything, and you still have more beetles in the winter than you do in the summer.
So you will have a lot of peaks then, huh? Like that?
Yeah.
You have graph like that, huh?
Yeah, I do.
One for beetle and one for the bees.
He can make it.
Different color.
Yeah.
Yeah, but 2015 is like backwards. Yeah. Is like the backwards year for some reason.
As far as the going back to the linear mix model, as far as interpretation of that, being that average bee number and temperature were the only significant ones, as far as explaining that, for average bee number, there was a positive correlation with that. The more bees there were, it was, I guess, significantly associated with more beetles, and that could be due to more bees being associated with a hive that has more resources and is healthier. As far as temperature is a weird one. I wouldn't have expected it.
Because what you just said is very important. What Jason was doing with the linear model is looking at individual hives versus the number of beetles and trying to find a correlation between the structure, the strength of that hive with the beetles. And normally the idea was the weaker hives would have more beetles, but it's actually the stronger hives seem to have more beetles, even beyond proportion. So this is what he's finding, like more bees, more beetles. Now, the problem is that overall, and this is what we were discussing, so that's a one-on-one, a single colony to the population of beetles in that colony. But what we're finding is if we look at the overall apiary, on a per hive number of bees, the lowest point for all colonies in that apiary is in the winter. And that's when you have the highest number of beetles. The relationship that maybe at that point, the stronger hives, even in the winter, attract more beetles is probably true. But it's hard to look at what scale you're looking. You're looking at a hive, individual hive versus the beetles like he did with the linear model, or is it worth also looking at the population as a whole of the apiary? So it could still be true in the winter, the stronger hives attract more beetles, but they're dinkier compared to the summer version. So that's one thing we were discussing, that we need to be careful what scale you're looking at it and what you're trying to associate individual characteristics of a hive or that big switch in population.
So in your linear mixed models, do you have any interaction terms or main effects?
As far as that, I think there was an interaction with size and average B number. I think those go together.
Size and average.
Average B number, yeah, like the number of boxes in a hive.
So you included that as an interaction term in the model, did you?
That one I included it in the model itself.
You did.
You're saying there was a correlation between those two variables?
Yeah,
statistical interaction.
I believe there was. I think there was, yeah.
Which makes sense because we're responding to their space needs. So you would expect that. You would expect it.
That's like a collinear.
Yeah.
Yeah, it's collinearity.
Yeah, but what Mark's asking is, did you include a term for any, like, allowing for any interactions between variables? So that's like when you include
cross.
Yeah. For that, I didn't. I don't think I specifically put that in there. I was thinking I could take size out because it was because it's so collinear with average B number.
Right, but still, you could consider things like the interaction between colony size and temperature or something. That is essentially kind of interaction.
Yeah.
Or other covariates like health of the colony and so on. You should how they interact with factors.
Okay.
Not across factors. What software are you using?
I'm using SPSS.
There has to be a way to keep interaction terms.
Yeah. I'll try to figure that out.
Jason, to address the problem of individual hives versus the impact of seasonality, right? Would it be possible to, in a sense, add some way of linking if that high number is happening during a certain season? Because a colony of 20,000 bees in the summer means something very differently. A colony of 20,000 bees in November. But it's probably not going to. I don't know. Would it be the temperature? I don't know if it varies that much. Yeah, I was really surprised at this temperature.
Yeah, the temperature was surprising because Hawaii, the temperature doesn't fluctuate. I mean, compared to
temperature and hive temperature.
Yeah, exactly.
Yeah, data from the NOAA station or data from NOAA in Waimanalo. Hawaii, in this climate, compared to like on the mainland, generally we stay from a range of around mid 60s, maybe at the coldest, to like high 80s. But it was correlated. Increased temperature was associated with decreased number of small hive beetles. As far as explaining that, a possible explanation I was thinking could be that increased temperature would mean that the ground is drier, and the beetle, from previous literature and other observations, the beetles do need a certain amount of humidity for a successful pupation.
Yeah. That's why you have to water your rearing. And this is 2015 rainfall. It was ridiculously higher than 2014, and so it was a big chunk. I forget, there were like 4 or 5 storms one after another in that period. So whatever happened was pretty atypical, and it definitely interfered with the requeening. And people have said that the small hive beetle is attracted to queenless hives. I don't know if I buy that, but I certainly think the instability and the population decline does put a colony more at risk. So I think, but we're just, like I said, we're just looking at the queens now kind of as a ricochet from looking at stuff that Ning has. So I decided, oh, maybe we can compare with Jason's, and then now it may give you a different outlook on things.
Would a different approach, I mean, I don't know if this would make sense, but if you wanted to look at the effects of different management strategies on small hive beetle numbers or different hive histories or something, would you be able to average, get your average small hive beetle abundance over 2016, because they're all experiencing the same precipitation and temperature, throw that out of your model and say, like, is average small hive beetle abundance related to average brood pattern, score, average size, average, percent of the time of I don't know how you measure status, but like, look at like the year long. I guess, look at things over that broad scale and see, like,
instead of on a individual hive by hive basis, like an apiary wide scale.
Well, no. I mean an individual hive basis, but not an individual sampling event basis. So like looking at like over that course of that period, was small hive beetle abundance on the hive level related to these hive characteristics? So number of times manipulated or a number of times harvested. It would be a different way to look at your question of this harvesting honey affect small hive beetle. So you look at like putting number of times harvested in 2016.
Like putting that into the linear mix model.
Doing a completely new one.
It's a different model.
Yeah.
And you're, I mean, just because sometimes like.
This is one case where I think meaning Jason and I need more help, because it's a pretty big data set that has, the unfortunate thing is that if you're not careful, you may get a positive for something that is ecologically or behaviorally meaningless. It just happened to, for whatever reason, So, but you want to try to tease out, like you're suggesting, like, how do you examine this in the best way to get something that could be meaningful?
Well, I mean, and the reason I was thinking this was because you brought up that point that some people think the number of times you manipulate or open up your hive affects small hive beetle. Could you look at that over the course of this season? Like, okay, some hives you open up, more frequently than others, or maybe that's not the case. Maybe you open them up.
Not for these, but the harvest possibly, like number of times harvest. I think it's worth even comparatively across years.
Across years might be
you wouldn't have all of those data.
Unfortunately, like bee number and brood pattern in 2016. That would be a difficulty.
I don't know.
Most of the time we only harvest one or two times a year. I can definitely try to look into that.
Okay. So most hives, so you harvest a similar number of times per year?
It depends on the year. Some years are super productive. I think when I first started, I think we had 3 harvests in that 1 year, and then recently we had a year where I don't think we harvested at all. Or we just harvested 1.
But like the reason, so if some hives were not harvested, that was because there was less honey flow in those hives. It's not like you just randomly chose. So that could be an issue then. Because your number of times harvested is related to other characteristics of those hives. Okay. Anyway, I like that. I like that you put together sort of an all-encompassing model.
Yeah, yeah.
So with this you will have 4 chapters, huh?
Yeah, yeah. So I was thinking.
Yeah.
You know what I was thinking when I was looking at the things you sent us previously was it almost looked like you just brushed through it and didn't make a huge effort to be synthetic in the discussions, for example.
Yeah.
You should really look at how other papers lay out their discussions, what they do, how it's laid out, how they refer to previous work and relate to the data. And so on. And other systems, it doesn't have to all be about small hive beetle. Other systems can be relevant, right?
Yeah.
So rather than just focusing on just your data, you have to make the discussions way more encompassing.
Yeah. That's another thing I was having trouble with. Trying to figure out what other bases to cover, I guess.
You can still write another chapter, very short chapter, like a 3 to 5 page chapter on combining combining the whatever you have got, all the chapters together, overall. So future perspective. Chapter name of the chapter like a future perspective. Something like a conclusion and future perspective or discussion. Overall discussion.
Overall, people often do that.
It can be between 3 to 5 pages only. Yeah, it's very short.
It's kind of, I think he needs to probably include in that something regarding the 2 main styles of beekeeping here in terms of large scale, because the honey producers treat their colonies, manage their colonies very, very differently than the queen producers. So the queen producers, to produce a queen, to make a queen, and he could explain the biology of that, you need to put a lot of bees in a very tight space without a queen. And you give them queen cells, and they need to be super well fed, they need to be super packed. And they don't even use anything against the beetle because the colonies are so tight. But that's a completely unnatural, very short temporary situation in a regular colony. So the honey producers have huge fluctuations in population size, or they move the colonies around, especially in the Big Island, from place to place, tracking food resources. But their colonies they're the ones who have losses because they have massive numbers and they can never keep them all super tight. So one of the things he could bring up, and is dangerous because you don't want to encourage people to just go, you don't need to trap, you don't need to do anything. But if you are really good at keeping a good balance between bee numbers and space, you don't even have to put traps. That's like a terrible little secret that we don't tell people. But because most people are not competent enough to do that. Scott's hives in the back of his house don't even have traps. They're just well kept for, and he has a good eye, and every time there's something happening, he'll take off a super. But this is like 0.1% of the people who keep bees who have few enough bees to do this and time to, or interest to keep checking them at that level. So the best thing you could possibly suggest is a low level, low intrusion, some awareness trap. The other thing that he needs to, I think, address is a while back there was a big fad for something called the flow hive, which was they gained so much money and what's that called? And
GoFundMe.
And those internet fundraising things to produce this hive, that the key issue is that you just crank a little thing and it can break the honey super, and then it has a spout. It's ridiculous. It's like having like a cap and for honey. So they were saying you don't have to harvest, you can just do this. It's so environment friendly, you don't kill bees. And some people in Hawaii have gotten, and they haven't done very well with it, because all these things that are happening in the hive, it makes you really lazy. It makes you think that the bees know what they're doing and then you don't need to do anything. And you just need to open the tap and get your honey, and it's totally okay. So it might be worth, although it's attacking a product, done with a little bit of caution, it might be good to include something about the need and the advantages to be in touch with your bees, because these things that we saw could be corrected with management. So it might be worth. The flow hive is the most extreme, and real beekeepers didn't adopt it, but it appealed to a certain audience that wanted to be in touch with nature, but easy enough that you can just open a tapping. It was really weird. Yeah.
So in this way, you can write maybe about 5-page overall discussion.
Yeah.
That will be it's just because otherwise these chapters are different.
Yeah.
And you pull together all the comments together.
Yeah.
What you have got, what is highlight your overall contribution to this research. So your research is more directed to beekeeping, honey cake production, not for queen production.
Yeah.
Yeah.
All right. Thanks.
Yeah.
So you can state this to difference at the beginning of the first paragraph, and what you have contributed towards the honey production through this research. You can highlight and add your comments and.
That's true, and you have your data for bee population, and I think you could play it up a little more because it does show the even though it's a categorical system, it does show the ups and downs. So if you were to refer to that data in the sense that you would get fluctuations, large population fluctuations in your bees, which is not what a queen breeder does. So we don't need to get numbers from them. We know how you rear bees. You keep a massive amount of bees in a small space.
So should you mention that somewhere that beekeepers are the two types, the queen producer and honey producer, somewhere in the introductory chapter?
Yeah, I could put it in the introduction
because you
as
want
well.
to wrap up with that at the to do that. Should you mention somewhere and your research? Yeah.
Okay.
Anyway, all this chapter will enhance the depth of your thesis. This fifth chapter.
Yeah.
And it will enhance the overall. It will give the PhD quality overall.
Yeah.
Yeah.
All right. Thanks.
Is there anything more to discuss today? Any question?
I mean, just with that point, I think you just have to make sure that your data actually back up what you're talking about. That you can point to what about your model backs that up. The compact, colonies.
Yeah.
Show your small hive beetle. Anyway.
Yeah.
Looks like you are going in you are making some progress. You are going in a direction, and I think this meeting was important. I realize now this meeting is important. I was not sure why I was coming today. Yeah. Okay. Okay.
Yeah, I think so.
It was not a waste of time. It was a useful time. Yeah.
All right. Yeah. Well, yeah. Thank all of you guys so much for coming today. Really
but now, whatever you have written, you have to follow through.
Yeah, I know. Yeah.
That is yeah. And you should have more meeting with the ITHAL.
Yeah.
Okay.
Everything you see, small thing you discuss.
Yeah.
Because you do not learn from anybody else. Actually, the most important person to learn it is hard.
Okay. Yeah. Thank you.
You have job interview.
Huh?
You have job interview
Tomorrow.
Good luck.
Thank you.
I see the advertisement yesterday. The job announcement. Job announcement. Yeah.
Seminar
announcement. No, the seminar announcement.
Yeah. You gonna be there? Yeah. Oh, no friendly face. Okay.
Well, all right. Thank you so much. Really appreciate it. Yeah.
Thank you so much.
Thank you. Your daughter is in England now, right?
No, she came back yesterday. She's in Animal Science.
Oh, she's in England?
She was at UCLA, but she headed LA.
Oh, she's in
I have a
England.
question.
Oh.
And she will maybe go to UC Davis.
Oh,
She just
gonna
taken
add-
that.
Oh, okay.
Can I ask you a
All right,
question about
thanks so much,
one
Mark.
little bit
Appreciate it.
of thing that I have? That
Yeah.
2 seconds, 2 minutes. So I'm gonna try in my talk to, talk about basically emerging infectious diseases, blah, 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 blah. But although I'm following a honeybee path and then I go into spillover, I want to make sure that people understand that we do a whole lot more that is outside. So at the end, one of the things we're doing, and I just wanted your opinion, is I'm starting a piece of work with archival chickens with a friend from used to be UACF, but anyway, with Amber. You may have a yeah, okay. She but anyway, what she was, it sort of coincided by chance. She was working in Salocopa from the basically a species ID because she think some of the mainland Medusa and is blah blah blah. So she was trying to straighten out who was where. Yeah. So
It's that native too?
no, it's Mexico.
Oh, it's Mexico.
But it's in the northern, the west coast, and it's here. So she was asking me for specimens to corroborate what she was seeing over there. Blah, blah. So she kind of like what's happening with that paper with the virus. Apparently, a box of samples is in a museum, and the museum is